Good morning, everyone. My name is Kendra Holland. I am currently a third year Ph.D. student in the bioengineering program. Today, I will be introducing Dr. Gaines, who is currently assistant professor of chemistry and biochemistry at Spelman College. She received her Bachelor of Science in Chemical Engineering at Michigan State University. Received her Ph.D. in Material Science and Engineering from North Carolina State University. She also received a postdoc from Georgia Tech and Emory, and also returned to Georgia Tech in 2007 to receive her third postdoc. Her main research focus is in microgels and hydrogels. Some current research projects are investigating the synthetic 3D culture microenvironment for biomedical application. Also, she studies the differences in biomolecular conjugation, distribution, and density that results from changing the physical and chemical properties of the microgel particles. And in addition, her lab studies how specific hair products affect overall hair health. Some of her past rewards are 3M Minority Engineering Scholar, graduate degrees for minorities in engineering, Jim Fellow, Procter & Gamble National Organization for the Professional Development of Black Chemists in Chemical Engineering, graduate student fellow, and last but not least, the inaugural Diversity and Inclusion Fellow from Georgia Georgia Tech, Georgia Institute of Technology. And she also received external funding from the National Science Foundation and Department of Energy and many more. Please welcome me in giving Dr. Gaines a big round of applause.
Thank you for that introduction. That was very comprehensive, and probably I wouldn't need to really go through my first slide, but because I always like to kind of introduce myself, but no problem. Thank you for that. Thank you for this opportunity to come and speak. This is a welcome back, I guess, also, because now we're in person. So definitely great to be that first speaker to do that. I'm going to tell you today about my research I've been doing at Spelman. And like I said, I'm going to start with a really brief introduction of kind of all of my diverse background that has come together to where I am now. And it's definitely great to be back at Georgia Tech, by the way. Wonderful to see all of the familiar faces. So thank you all for coming to this talk. So like I said, I'm going to tell you a little bit about myself. And then I'm assuming that many of you all are pretty familiar with polymers, hydrogels, and those types of materials. So I won't go in too deep there, but then I'll talk more about the actual work that I'm doing on these three projects. So first, who I am, like our introducer mentioned, I started at Michigan State in chemical engineering, where I did many internships at 3M. And then I went on to NC State and got my PhD in material science. So there was a degree change. And then I worked many postdocs. Now, when it said a postdoc, that would assume that there was just one, but there were actually very many of them. And it was not necessarily planned, but I really wanted to spend time learning about research and becoming a strong researcher. So that was my motivation for doing so many, one of the reasons. And like I said, I was at Georgia Tech, so I am so familiar with this room and this campus. So it's really awesome to be back, by the way. And then I made it to Spelman College, which is my happy ending. Well, at least happy ending in terms of my preparation, but now really happy beginning, because now I've started my lab. So I'm just going to mention a few lessons learned, because I think sometimes it's nice to know what your PIs have done and where they went, because you just assume that they just kind of jumped into where they are. And, now you can have a little appreciation for the steps that were involved. And as you can see, this is a windy looking road. And at times it really felt really windy, especially during my postdocs, but I made it. So what I learned, I love polymer materials still, they're still awesome. Use what you can from the past experience. So there's always something that you can learn or take from any experience, whether it's a tough experience or an awesome one. Sometimes you got to take a chance. When I started my first postdoc at GTRI, it was very different from a typical academic postdoc. And so I said, well, let's see how this goes. And it was great. I learned a lot. There were challenges for sure, but that's how it is with many jobs. And then plan, but roll with the changes. And I say this because I teach at Spelman College, which is a Black women's college, full of young ladies who have already planned out to the letter what they're doing from, the current age that they are right now to, 35. And I let them know that, hey, it's good to plan, but sometimes you got to change your plan. So you got to know when to do that. And then lastly, your path is unique. So embrace it. It's your story. All right. So I'm going to show you a bunch of smiling faces. These are most of the students that I have worked with. Most of them have graduated. One of them you might even recognize, Miss Gabrielle Brim. She was one of my first students that came to my lab, which was wonderful. Now she's here, about to graduate from biomedical engineering. So we're super proud of her for that. Most of these other students have decided to go on to medical school and dental school. Some are have gone to PhDs, and just a whole range of things. So super proud of them. And then also I introduced them to polymers because at Spelman College, we don't have a polymers class. So I'll be working on that. All right, so just in case you haven't heard much about Spelman College, it is right down the street and it's great because it is literally 10 minutes away. So it's always great because I can come back here easily and those that are coming can easily get there. It is right next to Morehouse College, which I know that's all right. I was expecting that. So Dr. Platt, Morehouse grad, we're so proud of not only our HBCU grads, but like our AUC grads, because that's at home, right? So yeah. So right next to Morehouse, right next to Clark Atlanta, right next to Morris Brown, and also another small seminary school. But with us, we're an all-female institution, mainly made of Black women students, so from the African diaspora, Africa, West Indies, definitely the States. There's about 2,000 of us, so it's much smaller. You can walk across the campus within 5 minutes. It's pretty nice, which is very different from Michigan State, where it definitely took me like 45 minutes to walk from one end to the other. Way different, but that's okay. We have about 27 majors, 8 of them STEM, and about 26% of our students are STEM majors. So we have quite a bit of STEM majors, most of them really interested in going to medical school and dental school. So a couple of pride points. We have a Phi Beta Kappa chapter. We have other types of like amazing scholars, the Gates Millennial, as well as astronaut and many others, Barry Gordy. More excitingly, we were just re-voted as the #1 HBCU, I guess, as of yesterday. So it's been a long-standing tradition. So it's been about 16 years that we've been voted in as that. So we're really proud of that. We want to maintain that and keep giving our students the education that they need. And also another cool thing is a lot of our students go on to advanced degrees, like I mentioned, medical school. So we're the #1 in terms of our students going on to get PhDs. So that's really exciting. So most of our students always go on to some other institution after they graduate. But then of course you can get a job. Some do that as well. All right, so bringing you back to me. So the Gaines Polymer Surface Chemistry Group, powered by 8 undergraduates. We're small. We are small but mighty. I combined the 2 words. So this is what we do. So I'm going to start kind of at a home base. Like we all understand hydrogels. So we're going to go from there, and then I'll get into the actual details of my research. If you were like me, you put in a hydrogel in your eye this morning. You put in some contact lenses, right? And what's great about them is we're able to engineer them so that they can mimic some sort of tissue, right? And so that's what a lot of us do. And they're great because they have mechanical integrity because of the crosslinks. They are breathable. They like water. So that's some of the characteristics about why we like to use hydrogels in biomaterials. And just to mention, the most important piece really of hydrogels, because you can see through this one in particular, is that it is swollen in a good solvent. It is water loving. It's mostly made of water. So it's swollen in a good solvent, which means that its mass, as far as the polymer chains and the crosslinks, typically take up usually less than 10%, but it ranges. Like I looked this up. I guess to be called a hydrogel, it has to be at least 10% water. But most of the hydrogels that we work with are much, much more than that. So yes, but the idea is you have these polymer chains and crosslinks that are expanded so that there's these large meshes. So there's space for things to go through, like water, for example, or growth factors, or possibly proteins. Difficult maybe for cells though, right? Because cells are usually a lot bigger. So that's one of actually the difficult things about trying to make a hydrogel for cells if you're expecting them to matriculate through. So hydrogels, but I said something about, or we said something about microgel particles or hydrogel particles. So these particles that I make are hydrogels in nature, but also colloids. So they're actual hydrogel particles. So what's interesting about colloids? Well, if you use mayonnaise or if you like ketchup, I love ketchup, or milk, all of these are colloids, right? And what's interesting about them is that they're able to sit at the interface between the aqueous phase and the organic phase. So like in milk, for example, the fatty acids are able to balance between the water because of the colloidal particles that are present. So they're at the interface. So that's the whole idea. And what's neat about them is that they're too small to see with the naked eye, thus the flashlight, but they disperse light. So that's why they're often opaque as well. So I said something about them sitting at the interface that might sound similar to polymers, right? Or block copolymers that sit in an interface usually between an organic and an inorganic, or an organic and a water phase, or just two different types of phases. So they can be like macromolecules. They behave like surfactants for sure, because they have a water-loving side and they have a water-hating side, polar and non-polar groups, just like surfactants. And then, of course, we said they're particles. So if you were to look at these on the macro scale, I just like to use this as kind of like a visual so that, all of us have held those little gel particles that smell great, right? They look very similar under the microscope. These are actually really big. These are about 2 to 3 microns in diameter. But you can see that being that they are filled with mostly water, they are really hard to see. So that's one of the challenges, but So also what's interesting about these is that just like any other hydrogel, they're responsive to external factors. So changes in pH, changes in temperature, changes in ionic strength. So what they do is they undergo this very drastic volume change, this volume phase transition. And if you all have worked with NIPAM before, you're already familiar with this for sure. And the particles that I make are NIPAM. So just for those that are interested. So here we are, 3 projects. The first one I'll talk about will relate to creating these particles or using these particles to create a 3D culture medium. And the way so I'm also going to show you pictures of all of the lovely students that have worked on these projects. And since I've had so many come through, I just think it's all right to show their pictures. So, so the way that I like to preface this project is the fact that thinking about cell migration. So if you have a cut, you have cells that are running to the rescue, right? You have platelets, you have fibroblasts, fibrin, I already said platelets, that are running to the surface to clot that cut, right? And so you have cells that are migrating. That's the idea. And we know that cells migrate because they have different machinery in their cell membrane that, like the signaling proteins and things like that, that help them to protrude out in different positions, establish a focal adhesion, and then they pull themselves through. So that's great for if we have holes that are small or pores that are small, and for cells that are still able to matriculate and move through. And here's what one looks like. Oh, I got to be over there to click it. Okay, well, I have a little movie. And this is one endothelial cell moving through the extracellular matrix. And so you see exactly this behavior as soon as I click this. Hey, come on. Well, what you would see is this cell moving through the extracellular matrix and having those protrusions come out as it moves around. And then I can't show you this movie either, but looking in 2 dimensions, so this is in 3 dimensions, only one cell. In 2 dimensions, like this scratch assay, you can see the cells moving in to fill in the space. So it's easier to model this in 2 dimensions, but 3 dimensions is very challenging. So I'm going to talk a little bit about the motivation behind this project and how it started, which would be at Georgia Tech. So we're looking at cell migration through for tissue regeneration. And so in this particular work, we had fibrin, and we had and we made hydrogels out of fibrin. And so as you see, we started with lower concentrations and moved to very high concentrations. High concentrations, you see, don't have very many pores. Might be difficult for cells to move through. So, which is what I'm pointing out here. And so what we decided to do was incorporate ULCs or BIS, so these microgel particles, and I'm going to refer to them as ULCs and BIS. There's only one small difference between them, but for the most part, they're NIPAM colloidal particles, and they're, as we see, infiltrated within the fibrin matrix, which is what it looks like here. So the dark spots are the colloidal particles. And so what we saw was that fibroblast migration actually increased when the microgels were present versus them not being there. And so I just realized, are you guys hearing me? Do I need to move this to a better location? Let's try that. Okay. So yeah, so we found that that fibroblast migration actually increased. And so then I thought, well, if I were a cell, which way or which medium would I want to be in? Which one do I think I could move around in best? Would it be the hydrogel, which would be like a jungle gym, the kid moving through the pores of the jungle gym? Or would it be the plastic balls, which I loved to play in, by the way, when I was this age? Would it be easier to move through a media like this? So I'm going to poll the audience. What do you guys think? Are you guys on the side of the hydrogel and the jungle gym? All in favor? Okay. All right. There's definitely some that, okay. And what about the plastic balls? Yeah, you know, and that was my thought originally too. But see, it's never that easy. But that's all right. So there's pros and cons to both. But what I started with first is assuming that cells are going to migrate faster through suspensions of particles than they would through a hydrogel film. So this is how we tested it. So we did this by looking at cell behavior or fibroblast behavior in collagen films and in suspensions of microgel particles that have been coated with collagen. So the first part was to characterize fibroblast behavior among the collagen 1 hydrogels. And so we cultured them on top and cultured them inside first to just see how they behaved. And on top, and I'm going to show you like a little illustration as to what we're seeing, because I think it illustrates it very nicely. But we saw that at 1 mg per mil and 0.5 mg per mil, that they were pretty okay. Cells were on top, they were stretched out, they'd established focal adhesions, they looked like they were doing fine, which is very similar to a tissue culture plate, correct? You know, I'm going to take a sip of water because I can see I need it. Okay, that's better. All right. So in 2 dimensions, we see that they're just flat, similar to on tissue culture plates. While when they were cultured inside in the precursor, the gel precursor before it was allowed to gel, we put them there and then we allowed it to gel. And we see that they look different. With the 1 mg per mil, that looks similar to the tissue culture plate or the 2D. But the 0.5, as we got to smaller dimension or smaller or lower concentrations, didn't seem to be doing so well. Maybe we could call them spherulites as far as the cells, but they were really more like singular in nature and very circular. So they hadn't established focal adhesions. They didn't look like they were doing very well. So at the lower concentration, we would also know that the mechanical integrity of the gel is going to be different. It's going to be a lot softer. And so that could be one reason why these cells didn't really like it. Now, what about the microgels? Well, I mentioned that there were 2 that I were looking at. They're basically the same chemistry, but we'll talk about that in a minute. And and I coated them with collagen so that the cells would recognize it. And, or not, we all did. And so the results came out like this. They both look pretty similar, but what you see is that you have very large spherulites, and they were kind of alive. I could see them moving around. Now, this was this is our first experiment. We must do this experiment again because we need to do some other things like change the media, take care of the cells, But this was encouraging to see because we saw that they were okay. So this is a good start. And so what we learned from this is that possibly for the microgel particles seem to do pretty well with the cells, but we may need to consider a combination, I think, because in terms of stiffness, because we see that as we get too stiff, they behave differently, just like we would expect. But we might need a blend so that they can really compare cell behavior. So that's the goal. Much like our fibrin composite that was created with this work originally. So that moves me on to my next topic, which would be looking at biomolecular conjugation density in microgel particles. So this was actually the first project that I started. And you notice that I have most of the students that have worked on this or are working on this right now. And this kind of came about by good old-fashioned scientific discovery. You do some experiments and you see something kind of weird. So I'll show you. So how we made these. So I mentioned that these were NiPAM, or poly n-Isopropylacrylamide. And so our monomer was the isopropylacrylamide. We also copolymerized them with a little bit of acrylic acid. And that's important because that's how we're able to functionalize them. It's also important because you'll notice that there's an ionizable hydrogen that's visible there. So that will come into play when we do more characterization. But I had a little video to show that it's a really fast process. It's basically a simple batch synthesis, and you have the monomers that are initiated by the initiator, and then establishing self-crosslinks pretty quickly. And then as the seeds have been formed, more and more monomer adds on, and it just continues to grow. And so that's how you get larger particles. So that's how that was involved. And this is actually started in Andrew Lyon's lab when he was here at Georgia Tech. So this probably looks very familiar for those that are familiar with his work. Now, I have a quiz for you guys. Aren't you excited? You get a quiz. Also, since I'm part teacher, I do a lot of teaching. I was teaching that way. Okay, so I'm showing you two AFM images. And one of them has particles that have cross-linking agent. And our cross-linking agent is bis. And so that's how I've been distinguishing them in terms of this 0% bis or 2% bis, which 0% bis is really ultra low cross-linked. But I typically say just 0% bis for simplicity because everybody understands that. So which one has the bis, do you think? The one is going to be on your left here, or the one on your right? How can we tell? Is there a scale bar to see? All right. So the scale bar here goes, it's not really from 9 nanometers to -9, but we know that's how it's read. So on your right, the height profile goes to about 18 nanometers, while on this other side, it goes to about 70 nanometers. So being that that's the case, which one is it? The one that is on your right that has the cross-linking agent? Okay, we're unanimous in that. The one on the left, does that have cross-linking agent? How does it look different? Okay, let's go back. This is where we know we got to go back and review. Okay, so we have this is the height profile, and these are the particles. The particles here look a certain way, while these might look a little bit smaller. Yeah? Yeah? Okay. And then we also see that we have a height difference. So this one goes only to about 18 nanometers, while this one goes to about 70. So all in favor for on the left, or no, on your left, that's what it's really supposed to be. Okay, nobody, we're still unanimous. On the right, yes, right, because the height is higher. So now you have, since you have cross-linking agent, it's increasing the density. So it's bringing everything closer. And so that's what allows them to sit up higher on the surface. So basically the 0% bis pancake against the surface. And so lately in the polymer physics world, we found that these ultra-low cross-linked or ULC particles behave slightly different than traditional microgel particles or colloidal particles. Instead of just being colloids, they also behave like fluid-like polymers. And so that's what this work is looking to investigate. So this is what they look like under the microscope. As you see, the 2% bis are a little easier to see because they're just more dense and they're smaller. And it also means that they have a different morphology in terms of its surface. So actually would have loved to talk about that earlier. So nope, I'm not going to do that because I will keep that there. Okay, so going back to the fibroblasts or the fibroblast work and the collagen that was coated, I told you that we were able to functionalize them on the surface. And I mentioned something about acrylic acid being the group that we would functionalize. And it also has this proton that can come off as well. But for the conjugation, in order to get the collagen on or any other protein, we use this EDC sulfo-NHS carboimide cross-linking chemical reaction. And some of you all may do that as well. We know that there's many ways to functionalize. Click chemistry, a long list. This is the one that we are studying or using. And so just to walk you through the process, because you guys may not see chemical symbols as often, because I know when I was in this room watching chemical symbols, I was like, oh boy, I'm glazing over. So I'm going to walk you through the scheme so we can see exactly what we're getting. So we start with this carboxylic acid that's on the surface mixed in with the NiPAM monomers. The OH, it pops off and, well, not the OH, the H pops off, and then we are left with this EDC that binds to it. That's a fast reaction. And then we add this sulfo-NHS ester, which is a slower reaction, but easier to protect from reverting and going back to the acrylic acid without the protein. And then we add our protein and we arrive at our final product like we want. And so the protein for the first project was the collagen. I've also tested a few others. And so we verified this first with NMR. And so what I would like you to see is that and so this is a big solvent peak, but here's the 2% bis, so the peaks for that. Once we add the EDC and the sulfo-NHS, we do that reaction, those peaks pop up. And then we remove the EDC sulfo-NHS and add the collagen. So we see this nice big collagen peak that's there. That was great to see. And then after washing it, it was still there. So the top is the spectra for collagen 1. So we were able to prove that we did have collagen on the outside. Now, some of you may say, but yeah, how much? How do you know how much? That is a good question. And that is the next question that I will answer. But first, I'd like to mention the 3 molecules that we have functionalized so far. There's more that we will, but this is where we started. So the collagen 1, we also used BSA. That was actually the very first one. And that's really just because it's highly available. So BSA, and then Alexaflor 488 Cadaverine. And so what I want you to notice is that I put the molecular weight for each of them. The Cadaverine is the smallest, about 600. The collagen is still on the pretty small side, about 1,000 Daltons. But you'll notice that the BSA is far bigger, about 66,000 Daltons. So we saw a difference in behavior, or at least conjugation density, between the BSA and the collagen. We also found that it was a little hard to characterize. So how we started out to do this first was by using the micro-BCA assay. And some of you all may have done that or used it. And the chemistry, the way that it works is you have a protein that's present, and it it measures via absorbance that concentration of the protein. And it's because it forms a complex with copper, which then reduces copper to copper 2, to copper 1. And then the copper 1 binds to the BCA dye. And so then we're able to measure the absorbance. Now, I'm showing you the results. And I'm also showing you, again, images of what the ULC microgels look like compared to the 2% bis. Now, we got different results. The 2% bis seemed to consistently give us more BSA on the surface compared to the ULCs. And that was what I said was surprising, because the ULCs, if you remember, are bigger. They're more swollen, they're wider, even though they pancake, they're still bigger than the 2% bis consistently. So why would a larger particle not pick up as much BSA or other molecules? And I thought, okay, maybe it's not real, but I found out that it actually was. Now, what you see is, in terms of the difference of the morphology of the particles, is for the ULCs, there's not the added crosslinker. So it's pretty homogeneous throughout each particle. While for the 2% bis, we've got a dense core. So if you have a dense core with dangly chains, now you have a definitive surface. And we think that that may be the reason why particles, or not particles, but within the chemistry, we're able to attach biomolecules more than in terms of the ULC, where they might just kind of slide right through or just slide right around. So like I said, I wasn't sure if these results were real. And the reason why is because looking here, this is 0% bis and 2% bis. And you can hardly see, but down here, we still see that there is less biomolecule on the ULCs compared to the 2% bis. But there's a control. And here are the controls. And you'll notice the signal for the controls is higher than for the ones that have been conjugated. And the reason for that is because of this peptide bond that's present in the NIPAM. And so that's interfering with the signal. And so that made me say, well, no more micro-BCA. We need to figure out a different way to characterize this, and actually, so I could convince myself that these are real results. But like I said, I told you they are. So you got to believe me, right? So the hypothesis is, like we said, is that the stiffer microgels, or the 2% bis, are more conjugated than the softer ones, the 0% bis. And so the experiments that we're doing so far, we did some absorbance, as you noticed. We also have done some fluorescence microscopy, and we've started to do some potentiometric titration as well. And so for the fluorescence, we had to have biomolecules that will fluoresce, right? And for the potentiometric titration, that was more so of a proof of concept to see if we can use this to actually compare. So with the fluorescence, you can see by eye, and also I have some smiling faces of people that you might recognize. Dr. Grover and I have been collaborating on this project over the last year, and it's been great. Her undergraduate, Sarah, has been working with us, and then Imani is the Spelman student that has been working on this project. And so we took these lovely pictures, and we saw just by eye, it looks like the 0% bis have a lower signal than the 2% bis. But the 2% bis are more dense. So is that really real? Yes, it is real. So we found that by quantifying the signal using the corrected total cell fluorescence method, we saw that there was definitely more on the 2% bis. We do need to create more samples and characterize them, but this was a good start and very relieving for me. So next we tried titration. And so titration when you took biochemistry or chemistry, you remember for acids and bases, when you mix an acid with a base, it neutralizes at a certain point. And it neutralizes at the equivalence point. So if you're looking for the whole curve, you're going to move past the equivalence point to see that inflection. And so what I assumed is that with these particles, we would also see an inflection. Because I mentioned, now in terms of with potentiometric titration, there has to be some charge that's present. And so that acrylic acid that has that ionizable proton on it, when that comes off, then we have charge. And so that's how you can get your change in pH from being at low pH to high pH. And so what we're seeing on my right are, so we conjugated EDC and Sulfo-NHS. I didn't do the protein this time because I just wanted to see if this was going to work. And the idea was, as we add more biomolecule, there should be less chargeable groups or ionizable groups that are present. And so that's what then that would mean that there would be less titrant that would be needed in order to neutralize it and to arrive at that equivalence point. So what we're seeing is the control in the black. Now it looks pretty, it seemed like it almost worked, but we need to do some more experiments. But we see that for the 0%, the inflection point is here when we added enough biomolecule to theoretically cover 50% of those acrylic acid groups. The inflection point seemed like it's a little bit higher, like it took more volume of titrant. But like I said, we're going to do some more investigation. But we saw with the 80% and the 100, we definitely saw a lower volume of titrant that was required. So we see those inflection points are further to the left. And so that was encouraging. So I think that we can use these results or this method to further quantify the amount of biomolecules that have been attached to the surface of the microgels. So just to summarize, we saw higher concentration density of the 2% bis microgels compared to the ultra-low cross-linked. We looked at BSA. We used fluorescence to measure the fluorescent signal with Alexafluor 488 Cadaverine. And then we also used potentiometric titration to compare the conjugation density when it's been covered with EDC and Sulfo-NHS. So that brings me to my last project, and one that students really like and are really excited by. And it was one that kind of came to me over time because I am a female. I have to do my hair. I have to figure out this conundrum, really is what it is, it feels like, because in a way, it's kind of like a material science experiment in a way every time when you do it. You don't know what you're going to get. You will wash your hair, dry it, and it will be completely different morphologically than it would when it's wet. And you would expect that. But it may not give you the properties or the desirable look that you're looking for. So then you have to go back and re-experiment. So whenever I teach the scientific method, I always use this as an example because we all can relate to that. Even if you don't have hair. I don't see anybody in here that doesn't, but there's always at least one person that doesn't. But you have facial hair, so that applies there. Okay, so anyhow, studying the surface chemical properties of hair, I said, they're fibers. There's probably a way of characterizing them further to better understand how to control them. And it turns out there is some research that has been done, but not enough. And so here I'm showing you an image of hair in its many different curl pattern forms. It goes from all the way to straight to kinky, coily, and curly, however you want to describe it. And traditionally also in the personal care industry, it's been looked at characterized just as three different main types. So quote unquote, Asian, Caucasian, and Afro. But we understand that those are more racial, sociological terms, but there's still phenotypic differences. So they're actually measurable differences. And even just looking at this image here at the cross section, we see that with the Asian hair, which is very straight, it's completely circular. While with the Afro hair, that has some wave to it, kink or curl to it, that it becomes elliptical. And so it seems as though this ellipticity might dictate, that could tell us a little bit more about the material properties. But it is a complex biocomposite material. And so it definitely is one that we can use to study. So just to tell you a little bit more about hair and what's inside each hair fiber or the shaft is you have a medulla, which is basically the core. But if you have blonde hair, you might not have a medulla. But I don't think you're missing it, just so you know. But medulla. And then from there, you have the cortex, which is the bulk of of the hair shaft. It is made of these cortex cells that are long, so kind of like bamboo. And for those of you that have worked with collagen, for example, you'll notice that the fibrous structure of collagen, which is made of, well, it resembles hair as well. And hair is typically made of, in terms of the protein, it would be keratin. So bundles and bundles of keratin, basically a keratin molecule, which then forms a helix with other associated keratin molecules, and then it extends from there. And so it's just bundles upon bundles of keratin. And this is what it looks like on the surface. Oh, and I didn't talk about that. So the surface is the cuticle, and it looks like little shingles on the outside. And it turns out that hair that's curlier has less shingles on one side of it than on the other. So there's many other differences in terms of curly versus straight hair that we're finding. Some more fun facts about hair and shampoo is that our hair is acidic on the surface, at least we hope so. If it's damaged, it might not be, but it's typically acidic. And so when you add shampoo, it's a surfactant, so it's attracted to the surface along with the water, and then it's washed away. So when we add conditioner, we're actually adding in positive charge. And you may remember that shampoo is negatively charged most of the time. Other fun facts, so shine is a thing that everybody kind of wants, or a lot of times in terms of desirability. And when all of your shingles are closed in your cuticle, you have more shine because you have light that's able to reflect easier than if they're open and damaged, actually. So it's one way of being able to notice damage. Also, a lot of intermolecular chemistry. So curlier, well, really all hair, they have intermolecular forces between the keratin fibers from one to another, as well, fibers, as well as within. So there's disulfide bonds, which are cross-linked, chemical cross-links. And then there's salt bonds and hydrogen bonds, which we know those can shift. For example, my hair is like this now because of the hydrogen bonds that have been reorganized. But as soon as I put water on it, it's going to revert right back to what it used to be. So a trick that we all use. Now, this came about, or at least in the early millennium, I would say, those that are curly headed people decided more so that they were interested in wearing the curls. And that's for many reasons. There was a awesome movie that came out called Good Hair by Chris Rock, and basically let everybody know that we're putting these really harsh chemicals on our hair. And maybe we should think about doing some other things so that we can get the look that we want to get. So Andre Walker came about. He is Oprah's hairstylist, and he came out with this little system on how to characterize the curl pattern of your hair, which is probably the most distinct thing that you can see. And it ranges from 1 to 4, and there's also subgroups in terms of the A, B, and C. So the idea is the lower the number, the straighter you have, and the higher the number, the curlier or the coily that you have, and as well as the letter as well. And what you'll notice too is that as hair grows out from the follicle, it also is different in terms of its phenotype. So the curlier the hair, the more it grows out at an angle. And that's just a few things. So there's been some work that's been done in terms of trying to characterize hair, because we thought, wouldn't it be easier if we had a quantitative way instead of just looking by eye? Because it kind of gets muddied, kind of becomes vague. And also, you might have more than one curl pattern on your head. I know I do. So L'Oreal did this study, and they had some parameters that they used to help quantify this. So they used what's called curl index and curve diameter, number of waves, and number of twists. So they surveyed, this was about 2007, so they surveyed about 1,000 people all over the world, and they collected their hair in different locations on their head, and they made these measurements. And so this first one is in terms of total path length. If you cut a piece of your hair and you put it down, and let's say it's 6 inches, then you're starting with 6 inches. If you stretch it all the way out, it might become longer if you have curly hair, but if you have straight hair, it might be the same. So they looked at the ratio, this curl index ratio. They looked at curve diameter as well, number of waves, and number of twists. In the interest of time, I won't talk about curve diameter, but if somebody asks a question, I can provide more information. But the number of waves and twists actually end up being about the same parameter, because they're counting, in this example, 3 different waves, but the twists are basically the number of times that it crosses the x-axis. So it's essentially the same thing. And what I noticed, at least from these results, so they used these parameters, they created associations using statistics without going too deep, and they came up with about 8 different types of hair that they're saying are in these different categories or families. 1 being the very straightest, and 8 being the curliest or the coiliest. Now, if you look at this data, you might notice that for 1 through almost 4, that it correlates pretty well. But when we get over here into the 5, 6, 7, and 8s, it looks pretty scattered. I mean, granted, I'm sure there's some similarities, but essentially it looks pretty scattered. And so the thought is that curly and coily hair need a better way to be characterized, or there need to be other parameters considered. And thus, here we are to do that work. Not just us, though. Like I said, not just us. There's a group in South Africa that they've been doing some of this work as well, and they came out with a paper that explained the systems approach for looking at human hair, basically to show that there's a lot of interdependence between each of these categories. So what I'm showing you, I know this is just kind of a busy slide, but you have this main network that's in the center with geometric character, biological structure, macroscopic properties, and physical and mechanical properties. And then they're showing the interrelation between these other main categories and physical properties. So the idea is that it's a really complicated system, just like anything else biologically, right? And so also here, they showed more about some of the other parameters that could be looked at in terms of characterizing everything going on with the cuticle, because there's several layers there, as well as some other parameters, including friction as well, which is a huge issue. If you've had breakage before, that's because there's friction that's involved. So the goal of this work is to take Andre Walker's system that's more qualitative and find some better parameters that can characterize these curly hair patterns or kinky hair patterns. And so I said at first we were trying to verify Andre Walker's system, but I think it's a good model to start with. And so these experiments were done at UMass with my student Imani. So she was in 2 different projects. And the parameters that we measured are on the right. I know I'm running out of time, so I'm going to keep talking quickly. But we're showing that the cuticles are different. So cuticle sheathing could be seen under dark field microscopy, and then also bright field as well. We used a similar parameter in terms of the total path length to measure or to measure curl index. And then we also looked at pitch, because if you have curly hair, the distance between one wave and another, one curl and another, is going to be different if you don't have, or if you have just an S curl, that's maybe just a few waves that are present. So we compared that. And so looking at this quickly, what we saw is that it did kind of correlate. Now, we didn't have any 3A or 3B samples, and this is all just really from one person. So these are all volunteers. Some of them, you'll notice I'm up there, and my daughter's up there, actually. So, but what we saw really is that the contour length, just like in the L'Oreal paper, from all the way down from this 2, wait a minute. We see categories in here where the 2s in terms of contour length are longer, well, the 2s, then the 3s get smaller by a lot, and then the 4s get even smaller. We also saw, so the difference between contour length and 3 centimeters and contour length altogether is if we just added more length to it. But the idea is that we got similar results, but we're going to do some more studies. And also to mention too, that when you have curly hair, so we did a little bit of mechanical studies, we saw normally what you would see in a stress strain curve, which you see tensile strength, and then you see an elastic region, and then it breaks and it fractures. But there's one additional piece to curly hair. It's the uncurling force. So the South African group coupled that along with the 2 region, the elastic region, and they said that was all a part of the tensile strength. But I think it's something that could be compared. So we saw this, what's called the toe region, so that's the uncurling force. So you can imagine with the curlier samples, it's going to be longer than it will be, or larger than it would be with straight samples. It's not going to be any toe region. And we saw that. So first I'm showing you fracture force and modulus. And modulus doesn't include that uncurling force. It's just the actual fiber strength. And we see that going from 2A to 4C, that the fracture force definitely decreases. So it breaks easier with the curlier the hair. And we also see that with tensile strength, I don't know if we can conclude a lot yet, but there seems to be a drop between, this would be 3A, or Lacia's, 3C, and this is 4A. So 3C actually, I think, has some of the most interesting properties because it has basically these big loops of hair with big curls. And so there's a lot to see in there. So here's some more of the results. And like I said, Orlacia's hair is the pink one. And so we see that there's a large elastic region, just like there is for the 2C as well. The 4s are down here, and they're smaller, but you see with the 3C, this large toe region that's present. So that we see indeed there is a large uncurling force that's present. We saw this before. Okay, then we have also, we've done a little bit of I can never say this word, but this, some words, I don't know. Anyway, but what's nice about this is now we're looking at the hair fiber in 3 dimensions. So now because in 2 dimensions, you can see where it goes up and down and maybe twists and goes across the x-axis, but there's more to it in 3 dimensions. And so we're using this technique as well. Thank you all for coming and paying attention. These are my acknowledgements. I have all my students once again to show, as well as all of my collaborators, and I'm thankful for my new funding, which paid for the conjugation density project. And at this time, I'll answer any questions.
Thanks, Dr. Gaines, for that interesting talk. Now I'm going to open the floor up to Q&A. Could you please raise your hand and I'll bring the mic over.
Hello, Dr. Gaines. Great to see you again.
Good to see you too, Dr. Platt.
Question about the last part really is a simple question, I think, but the follicle shape, how do you identify follicle shape? And then are there any hypotheses about why the shape of the follicle induces the different curl patterns?
Yes, that's a good question. So There was a paper that characterized more of that, the curl as it comes out. So it's called, basically, in your follicle, you have living hair, and then as soon as it comes out, it's dead. And so while it's in the living, like, transition phase, that's when it is maybe being programmed, if you will, to be curly or not curly, based on probably your genetics. And so as it comes out, it's basically, the curl is the result of whatever is being done in that, I guess it's really like a cross-linking and a polymerization phase while it's in that living space. So it's kind of interesting, yeah. And there's more too, in terms of like the turns, why it turns. There's some physics as well involved. Yeah, good question.
Thanks. That was really, really interesting. I was wondering, for the conjugation work, if surface adsorption also played a role. So the chemistry is one thing, but the protein or biomolecule conjugating has to kind of get there in the first place before it can react. And so I wondered if having that well-defined surface provides
More
sites, right, for physical adsorption, as opposed to the squishy ones where there's no actual surface, right? As you described, it's just kind of more like liquid.
Right. That is a great point that I didn't think about. And that is probably why, is that surface, there's just that ability for it to find the surface, and it can't find it with the soft ones. So that might be a way to just kind of think about it as I'm moving forward. Thank you.
Thanks. Thank you for the talk. Going back to the hair studies you're doing, you mentioned that it's possible to have like different coil types along your head. So how do you, when you're taking your measurements with your lab, do you take different parts of your hair, or do you stick to one side of your head?
That's a good question. Now, actually, how we got the samples might be even, it affects what we're going to see. So really, my samples that I showed is hair that came out in my comb or my brush. So it probably will behave differently than if I took it along different parts of my head. And I have no idea, where it came from as we did the combing. So there's already some physics that it's been susceptible to. But then in general, hair has so much history. We've done so many different things. Maybe we colored it, maybe we permed it, maybe we added heat, maybe we didn't. And so all of those pieces of history play into the physics. Yeah. Other questions? Anybody characterize their hair yet? You guys know what curl pattern you have?
I have one crazy question. Has anyone made like a synthetic system to grow like pure virgin hair in a tissue dish so you don't have the chemical? Have you seen that either?
So I found some cells. I found some cells a while back, and I thought I'm gonna get them because we need to grow some virgin hair somehow. How that will work, your guess is as good as mine right now, but I'll let you know. Yeah. Because beyond that, how to find virgin hair, I was thinking probably like boys in elementary school, because there's nothing happening to them, They still wash their hair, right. But that's the most that's going to happen. It hasn't been colored yet. It hasn't been, processed yet, most likely. So yeah, as long as we don't care about length, right? Just simple. I guess I should have thrown my son in here too. His hair, it was going to be different. Other questions? Well, thank you all so much for this opportunity. Thank you for coming as well.